a couple. Recording in progress. Very good. So we're going to have a couple of very interesting talks. Wendy Chung from Columbia University is going to talk about the COVID Recovery Corps, and we're going to have a educational project or discussion presentation about browsing research projects, institutions, and researchers using the enclave object explorer. And it's remarkable. We're well into our second year, and we have 61 sites have released data into the pipeline with a combination of models, and 223 sites have executed the DUA, and so statistics, we have 2.1 or almost 2.2 million COVID positive cases. It's really quite a compendium at this point. 56 sites with over 7 billion rows, over half a billion clinical observations, 3.5 billion lab results, and so forth. So it's things are moving along. It's really quite a remarkable compilation. So now, without further ado, I'm going to hand it over to Wendy. So you're going to share your screen. So I'm going to stop sharing.
Okay. So, okay, hopefully everyone can see this now on full screen. Actually,
need to flip the display.
Yeah.
Yep.
Okay, give me just a second. Sorry about this. Okay, so are you seeing this now on full screen?
Looks good.
Okay, sorry about this, not my usual computer. So I'm Wendy Chung. I'm at Columbia University, and I'm proud today to talk to you about some of the things that may complement what you've been doing. And I'll say unabashedly, if there's anything that's useful that we've built, I think I can speak on behalf of Sage Bionetworks, at least I can speak on behalf of myself. Anything that we've built is available to the community, so I hope others can make use of anything that we've done. So I'm in New York City at Columbia University, and we got hit really hard early in COVID, and with the very multi-ethnic cohort that we study at Columbia, or that we serve rather, it was very clear to us early on that there were differences in terms of who was getting sick and who was coming into our hospital, who was critically ill, who was dying. And as we started looking at this, it was very clear that there were disparities, not entirely clear what all those differences were in terms of why we were seeing those. But there were things that over time have become more obvious. And I won't say it's just at Columbia, but we were sort of early seeing this. And I hope we're able to shed some light on some other things going on. These are data that came from the Columbia study. I'll be getting into survivor or COVID Recovery Corps in just a second. But as I said, when we looked at this, there were clear differences by race, ethnicity. I don't think they were just in terms of, I'm a geneticist as well, just based on genetics of that, but had to do with social determinants of health and access to care and population structure and occupations and exposures and lots of other things. But within this, of course, you all know better than I how you can do really elegant research by having well-characterized cohorts clinically and being able to see signal even for complex multivariate contributions. So as we were doing this, and as we were starting to see, as I said, some of these contributing factors that now how even a year ago, people could have said very easily in terms of the patterns that we were seeing, we saw risk factors both in terms of who was getting sick and then also who was recovering. And as I said, it was clear in terms of, for us, a Latino, non-white and Black population specifically at Columbia, how much difference we were seeing not only in hospitalization, but in disease severity, whether you looked at mortality, intubation, time to discharge, any number of metrics. And again, in the very early days, this was April, May 2020, we were already starting to think about, for those who did survive and those who made it to discharge, or in fact, those who were never admitted, trying to think about what this was going to be like longer term. And so already starting to think about the recovery aspect. And again, this was April 2020 as we started thinking about this. So we were seeing, with this, individuals, as I'm sure all of us are now aware, both with mild disease, but very, very severe disease. And as I said, it was really uncharted territory. I think we were, I'll be honest, scared to death in New York in terms of what this was going to look like long term. And we were starting to hear and see ourselves through even ourselves. I have to say, we as healthcare providers were some of the ones that were facing some of this ourselves, what that recovery process was like. At the same time, I'll give a lot of credit. The patient community was also self-organizing in terms of themselves to be able to understand and to share. And in the era of social media and things like Facebook, able to share experiences and I think support each other. And so, as now everyone knows, these different terms have been used, but long haulers or COVID recovery or other things have been described for this. But what we were seeing were certain people were bouncing back right away. I'll say especially some of the kids, the younger children. I'm a pediatrician. We're bouncing back right away, but some of the other special middle age and older adults, but even some very healthy young people not recovering as quickly as we might have expected after something like the flu. So we came together to think about what this recovery process would be like. And in particular, some of what we were concerned about was filling a gap perhaps between those who weren't coming into our healthcare systems and were just out there in the community. Some people who were in our healthcare systems, but perhaps not re-accessing healthcare either because of access to care issues or other things. But we understood, and I, for instance, myself know what our EHRs can provide us, but clearly understanding the gaps. And even for patients who were accessing care, whether or not there were symptoms or other things that weren't reported in the EHR. And so again, trying to fill that gap, trying to realize that not everyone can do everything. And I specifically took the strategy of not trying to cover the things that were in medical records, but getting permission to access that information and supplementing it. So thinking about participant reported information that would be able to supplement EHR or other public health data that would come from this. So with this, one of the challenges that we had, and again, you've got to think about the time frame for when we started this in April 2020. We didn't have enough COVID testing. So we didn't have enough, being able to do PCR and being able to do nasal swabs and do this. And so we had a lot of people out there who thought they had COVID, but it was hard to know for sure. They could have had some other viral illness, and being able to understand and very well define who actually was sick and sick with COVID, that is, became a really high priority. And in New York, we had the idea that if we got sick first, in other words, if we took the first hit, we would also be able to be the leading edge in terms of seeing what the longer term manifestations were, and perhaps be able to alert others in terms of things to watch for. So that was the thinking in terms of being able to set this up. Within this, we did try to do everything we could in terms of making sure this was accessible to a very broad community. So materials, or you'll see some of the examples in a second, but very much in terms of English and Spanish for the community we serve, acknowledging the community so that individuals participating and would feel connected both with our study staff as well as the visual imagery that we used. And we wanted people to get something out of this. And again, part of what was gap that we were filling from the diagnostic side is that initially, without sufficient COVID testing, people were interested in understanding their antibody status to know if they had evidence of prior infection. So in other words, being able to have a free test where they could see from initially a blood sample, and as I'll show you later, a blood spot, being able to see if in fact they had been exposed and were recovering from the virus, so that we could be able to say for sure, in fact, they were part of COVID recovery. That was a definition for us in terms of understanding that. We did, and we're willing and still are willing to take people who can actually demonstrate that they were infected because they've got a PCR test. And as we've had participants that have registered more recently, that's been easy enough for them to do, that upload that test report and review that. But as I said, early in the process, that wasn't possible. So this was somewhat challenging to do because we were literally making this up as we were going along and iterating on the fly with this. And that was fun. It was challenging. I'll also say it's been ebbs and flows, I think, as this has swept through the country and as other political things have happened. But when we finally launched with this, what we were aiming to do was initially to have, again, a free, accessible test that could be done at home. So we had a lot of quick validation to do because the idea for an at-home test for antibodies was that you could do a finger stick. You could take a blood spot and put it onto a filter and be able to dry that for overnight, basically, send it back into us in the laboratory, and then we could extract the antibodies from that, be able to report that back out through our clinical laboratory. So again, this is not a research result, but through our clinical laboratory, and get that back out to participants. That would help them to know that they in fact had really been infected with COVID. And then it also helped us in terms of our case definition. So with this, it was not easy to do because I'm sure most of you, or maybe none of you, are laboratorians, in the sense that in a laboratory, having to go through a process of developing a new test and validating it and have a gold standard for comparison, and then finally to get it past New York State. So all of this is actually a multi-step process in terms of doing this. And we were trying to do this as quickly as possible because some of you know that the antibody levels will actually come down over time. And so if you wanted to do this, you needed to be able to get people in a window where we could still, through serologies, check those antibodies. So initially going through a phase where we had individuals where we were collecting blood for a gold standard, at the same time also collecting a finger stick from them so that we could then do the comparison with what we were doing. And I won't give you all the details, but Eldad, who's a colleague of mine in pathology, and Alex Ray going through a lot of work in terms of validation, developing the methods, developing the sensitivity, again, with extracting the antibodies from a relatively small blood spot. All of this in the same time that obviously people were quite afraid in terms of being able to even go out. You know, people were still sort of huddling and sheltering in place in terms of doing this. But on the other hand, also quite interested in being able to contribute something to their community. So after, being able to do all of those things, then being able to go from something that was based in New York City to then being able to open it up to a broader community and thinking about nationally if there are other individuals. And of course, this was during the same time that all of us know that, it wasn't confined to New York, very quickly spread, and being able to allow this to spread then on a national basis. And so this online platform that we have, in fact, is nationally available for anyone in the US to be able to sign up, as I said, in either English or in Spanish. So this was, and I give a lot of credit to CZI, to Chan Zuckerberg, for helping with us from just the professionalism in terms of what this was. This was done very quickly, but I think done very well, being able to take members of the community and really get both their input, being able to get the visual imagery of what we were seeing in New York, and really reflect our community. And doing this, the campaign in terms of recruitment included everything from being in our churches, being in our community centers, being at subway stops, being in our medical center, and then online in terms of doing Google Ad and Facebook in terms of being able to reach out. It was a real, I think, the study staff that we had doing this, again, were bilingual and very much from our community. And I think we're very much accepted and appreciated in terms of especially helping individuals who might not have been so savvy. This is an online platform, might not have been so savvy about doing things online. And one of the things that we noted is, despite, I think, a huge amount of support for individuals to be able to participate in multiple ways, it is interesting, I'll show you some data in just a second, but how we still ended up recruiting what is for me a pretty typical population for research studies. That is, individuals who are relatively self-selected. I think we got a wider diversity in terms of socioeconomic status, but not as much diversity as I was hoping we would, to be quite frank, in terms of our racial ethnic diversity. So with this, we did limit it to adults. So we don't have children in this study. This was in part, as I said, because it's an online study and we needed to make the consent and the workflow relatively straightforward. It was intended to be only individuals who had COVID or thought they had COVID. At the end of the day, for people who thought they had COVID, it was over 90% who really did have COVID. So we didn't have too many people who were false positives, so to speak. And that was even true of the people who didn't have nasopharyngeal swabs or PCR tests. Within this, we had this set up so that individuals, as they're doing this online, were not compensated for this. This was done in the spirit of really helping the community. So unlike some other studies that I do, they didn't get any gift cards, they didn't get any compensation, but the idea was that they could be able to compare to see how they were doing with other individuals in the community. And the what's in it for me, so to speak, was also being able to get their COVID results back, their serology results back. Within doing this, the idea was that there's a baseline survey, being able to do the consent, the registration, and the first part of the surveys was still about 30 minutes. I will say I particularly like our consent as we did this. I thought it was both very transparent, the infographics really great, and again, Sage was fantastic with that, and also is interspersed with a few pauses, not in the means of sort of a quiz, but it really ends up just being a pause with a simple multiple choice question that we've got interspersed within the consent, just to emphasize the important points. Within this, as I said, individuals weren't required, but if they didn't have their nasopharyngeal swab test, then could donate the sample, and being able to get that done in terms of the testing. Those samples have not been used for a biorepository, so it was not for the purpose of making a biobank or biorepository or doing anything further from that. It was really for the purpose of individuals being able to get the results in terms of their serologies and the case finding. We tried to make this easy, self-serve as possible, in terms of, individuals, if they needed to come in and give a blood sample, being able to choose their own time and being quite flexible, both, as you can see here, with weekend hours, and really opening it up to make it flexible for the community. As we did this, I don't know how many of you have ever done this, but it's fairly straightforward in terms of being able to get a blood spot. That's something that actually, based on this, we've developed other assays and other things we can do for at-home testing based on blood spots. But this proved to be actually something relatively simple. We have nice videos for people to follow in terms of collection, nice instruction cards, and have had actually relatively good compliance in terms of actually giving us enough blood to be able to do all the assays, and very few repeats that we've had to go back and get. So that was quite effective. As we're doing this, we have had this idea that we wanted to relatively quickly share information back to the community. I will say that over time, I've wanted to make sure we had enough data, and data that was meaningful enough to be able to share back. But we've done that both in terms of, we have a portal for people to come into to continue answering not just their baseline surveys, but they get longitudinal surveys over time. And from that can log in and also see the results from the study writ large, and be able to feel like they're part of that learning process. As I said, the other part that we do is within, they get a confirmation out from our laboratory that also tells them their test results are ready. This also is available through their portal, so they can always go in and be able to download a copy of their test report. And with that, then in terms of education, because this is a clinical report, I made a video along with our infectious disease doctors being able to talk about the differences between different tests, PCR tests versus serology tests, and help to explain in terms of what this means. And based on the customer service email and phone number, we have actually very few questions in terms of this. So either our folks are just really with it, or they were easy to understand this, but this has proven to be relatively effective. And again, I say this mainly because if there are any parts of any of what I'm talking about that are available, either COVID studies or other things, I do hope that people learn from this as a process. So in terms of individuals that have filled out the baseline questionnaire and fully enrolled in part of this study, a little over 1,100 individuals. In terms of the demographics, still largely a white population, more females than males, reflective, I think, largely also of other cohorts that I've had in terms of the difference, the sex difference. We did have a little bit more in terms of spread, in terms of at least income levels. Again, reflecting what we see in New York City, and individuals, even though the income was, I think, more representative, the educational attainment a little bit higher than the average New York City population. So to get to probably some of the most interesting parts of this. So these are symptoms that were reported at baseline. Individuals could report more than one symptom at baseline. I'll show you in just a second when we split this out by sex, but I think this is similar to what we've seen with other studies. I don't think there's anything sort of too earth-shattering here. Fatigue, almost everyone basically had fatigue, and the things we hear in terms of brain fog, headaches, myalgia, shortness of breath. I think those are very similar to what we've all seen with others. Greater specificity in terms of loss of sense of taste or smell, and then other things that we see. Interestingly, some of the things that you've probably heard but are definitely real were things like the hallucinations. Thankfully, other things like seizures, less common in terms of us seeing them, but I was actually surprised at how frequent the hallucinations were. Splitting these out by sex, so the same data, but now splitting this out by males and females. Again, there can be a bias in terms of who decides to join the study, but we did have generally more symptoms, or more frequently symptoms were reported than females than their male counterparts. But I would say that these were very similar, not huge differences in frequency between either ones. And I don't think there's really much in terms of sexual dimorphism that we're seeing here. More importantly, as I said, we had baseline, and then we've had follow-up surveys. That we have what I call long form and short form in terms of the follow-up surveys. The longer form is more of a systematic way of being able to see what symptoms people are reporting. And then a short form is really literally, I call it a micro survey, but it's just being able to see if there are new diagnoses or major new symptoms that people are having that they can report in, so we can get a sense of that leading edge. In terms of this initial symptoms in blue, and then in orange, the longer term symptoms. And the point is that people are getting better. So for most individuals, in terms of time difference between when they did their baseline and when they did their follow-up survey, is at least 6 months for most individuals. So there is time for recovery with this. Still, though, I guess most notable is they're still continuing to have significant problems, a little over 50% of individuals with fatigue. Of course, there are a lot of things going on in society, so I won't say that everything is due to COVID in terms of this, but that fatigue factor and that myalgia and just feeling kind of run down, still quite common in terms of symptoms in one sense or another. The CNS symptoms in terms of headaches, confusion, brain fog, even hallucinations persisting a little bit, those are still there in terms of what we're seeing. And even, things like recovering taste, smell, although they're definitely recovering. Impressive to me that there are still so many individuals that still continue to notice differences in terms of that. In addition to that, we've been taking a look at new diagnoses, so things that are actually diagnosed by their doctor in that time period. Again, some of these are relatively low frequency along the bottom, but the thing that's most impressive, I think, are the psychological and behavioral and psychiatric diagnoses. Again, not saying that this is all COVID, a lot of other variables changing at the same time, but between anxiety, depression, a significant burden in terms of both the number of individuals affected as well as what they're telling us by how much they're affected. On the other hand, I will say the good thing is, almost half of them have nothing new to report. So that's good that those folks are continuing to recover. And as I said, one of the things we've been looking closely at are things that may be infrequent, and especially if they're very infrequent, maybe nothing. It may just be, the background sort of noise that we see going on. But we've been looking for things specifically that follow the pathophysiology of what we see, either in terms of what cell type or tissues are affected by the virus or by other things that might be related to the host immune response to the virus. And so within this, I'll just point out, and again, I think this is very, very early, but some of the more unusual things that we started seeing that could be related to some vascular problems, pulmonary hypertension, some lymphatic issues that seem to be associated with lymphatic changes and lymphatic obstruction, as well as things, for instance, even like dental things and periodontal things that may be related to vascular issues in terms of even going to the gums or the teeth. With this, we also had the ability, thankfully, to get vaccine data. So individuals that were being vaccinated. And very early on, I was initially surprised, although it was borne out by others as well, that there were individuals who thankfully were improving actually after vaccination. Within our group, about 2/3, even though they knew they had been infected, so again, case definition is that they definitely were positive, were vaccinated, and of those 2/3 vaccinated, about 2/3 improved. So that was not everyone, but it was good to see that some people improved. So just wrapping up in conclusion with all of this, to say that as we've been looking at this, we'll continue following this cohort over time. It has issues, I'll be the first to admit, in terms of the, who self-selected to come into this, but they do provide some opportunities to be able to see longer term, either what symptoms are not resolving, and in particular, some that are maybe perhaps more unusual diagnoses that may give us some ideas about how to continue following others over time. I do hope that individuals are able to get back to whatever their new normal is, but we have the sense that there is going to be a long-term burden associated with this. So just at the very end, I want to acknowledge all of the people that were doing this. They really pulled up their sleeves and, in times that were quite trying. In particular, Survivor Corps has been an amazing patient organization to work with, CZI, our funders, and Sage Bio Networks, who are incredible. So I'll stop there.
Wendy, thanks. That was a fantastic talk. Really, very, very interesting. Questions? I'll start out actually. So you were talking about symptoms after vaccination, just to make sure that I'm clear on what you're saying. So these were patients who were not vaccinated before their infection, that this was after so they were not breakthrough patients. They got COVID and then they got vaccinated and then they got better. So I guess the obvious question is, were you able to do any sort of propensity matching or any kind of comparison? People who weren't vaccinated, did the people who were vaccinated seem to get better faster than people who weren't? I guess it's difficult because, of course, most people did get vaccinated. But do you have any senses whether there's any causal thing going on?
So we haven't done the analysis yet. We still are early, I think, in terms of doing that. So timing in terms of this, the data in terms of how far out we are after vaccination is still not very far. And so I think in terms of being able to see those differences, I just don't think we've had enough time for that to develop. But you're right in terms of the sequence of things. Most of the people that were recruited for this were sick very early and then got vaccinated for those who did, sort of January, February, March 2021. So, and everyone was sick and initially infected with COVID much earlier in 2020. So I think we'll see over time in terms of whether or not, and it's going to be multiple variables that we're going to have to control for in that. I do worry that our cohort may not be of sufficient sample size to be able to see those just in terms of the power.
Yeah, that's a fascinating. Do you have any speculation as to whether vaccination might have any positive effects on people who might be developing, post-COVID syndromes?
So it's complicated, at least from some of the things that we saw them reporting. I also wonder how much of it is, I think a sort of outlook on life, I guess, is one of the things I'll say. There were definitely feelings of just feeling safer and perhaps less anxious with some of these. So I think that's kind of an amorphous thing. I think what we'll see over time are some other factors in terms of diabetes, some of the cardiovascular complications, and those I'd like to also be able to see whether or not they're affected. Our frequency of those is too low, and we don't have enough time to have enough events to be able to say really what that outcome, what those differences are.
Great. Okay, so we've got a question in the chat from Zia Jing. I'm curious how to define what symptoms are related to COVID-19 during the follow-up period. Here's another question. We'll start with that one.
Can you say that one more time? How to define which symptoms are related to COVID-19?
Right, during the follow-up period.
Oh, well, I mean, in terms of ascribing attribution, it's, I don't know that you can directly ascribe attribution, right? I mean, you really need to be able to look at in terms of a comparable cohort of individuals who are, essentially what the rate of new diagnoses of X, Y, and Z are in terms of doing that and trying to control for as many variables as you can that are also contributing to those things. So I will say, as I was saying before, when you start getting complex in terms of doing these, I think there's only so much you can do with a sample size that's the size of what we have right now.
Right, no, I understand. And then another question from Zia. Let's see, about 2/3 improved after vaccination. What are improved symptoms? Is this result of self-reported report by participants?
Yep. So the things that we were seeing mostly in terms of improvement of symptoms were, like I said, anxiety, brain fog, sort of fatigue, lethargy. It was those most common symptoms, but those that we're reporting in terms of improvement.
Great. And then Hannah Blau asks, did I see that about 1/3 of those who got vaccinated found that their symptoms got worse? Or just didn't get better?
They did report things getting worse, although one of the things that we need to get a little bit farther out is our sense was based on timing of when they were reporting this and how long they were reporting this, that it was more acute in terms of their symptoms getting worse. And so I think there were issues in terms of, we were seeing everything from headache, myalgia, fatigue in some of those individuals. And again, it was only about 1/3 of them, but they did see sort of an acute time period, and acute meaning oftentimes for a week or two after their vaccinations, and then sort of coming back to baseline for the most part.
Great. Well, thank you. That was really a fantastic talk, and really appreciate it. So now Shawn O'Neill is going to talk about Object Explorer browsing research objects or research projects, institutions, and researchers. Shawn, I assume you'll be sharing your screen as well.
Yes, I will. Thank you. Can you guys hear me okay? Hear me and see my screen okay?
Yes, we can, Shawn.
Thank you, Ken. All right, so I wanted to show something. This is just a little bit, I think, of fun, but I'm going to show kind of two things. The first one is I wanted to show a little bit of the backside of what happens in the enclave, so that you guys have a sense of a little bit of how the sausage is made, as it were, for some of the enclave-based tooling that we have. And then I wanted to show you guys how to use it in order to do something fun, which is to explore how projects and institutions and researchers relate in N3C. So to start with that, I'm going to go to the training portal, which is under the training material button here, because this is my opportunity to tell y'all yet again about the amazing things we have in the training section, especially the training portal, which is stores a bunch of N3C specific training modules related to, you know, things that you can do in the enclave, some OMOP, all that sort of stuff. And this is you know, so we've got some training modules here, and this is an interface that we can show these. And if I were to click on one of these, such as our Enclave users group, which is meeting most Fridays, we can see that there's sort of some information here about this particular item. You know, the title of the training module, a description, some topics, suggested background. And then over here on the right, we have some resources, which are links to actual resources that we've got going on in the Enclave users group. So like meeting recordings and an FAQ and that sort of thing. And what's interesting about this is to actually look at how this data is stored and used in the Enclave. And so I've got two tables open here, and these are just regular old data tables, much like we've all seen so often. And really here, each row is representing, this is the list of training modules, and each row here represents a training module, right? We do this a lot where each row on a table represents something, and then each column represents something about that thing, right? And so the Enclave users group has module ID 19, it has a description, maybe it's got some topics, and these are just like, you know, text fields and things. We have a date field for the last time it was refreshed, where that's out of date. And I got to update that. Suggested background, stuff like this. Some of them can be, you know, numeric kind, some of them are strings, some of them are dates, and so on and so forth. And then the other one that I have open is the module resources tab. And this is basically the listing of the resources that are affiliated with a particular module. So module 19, which I said, which is our Enclave users group, has a bunch of different resources. And each row here is a resource associated with that particular module via this foreign key. So the title is, you know, some webinar and example resources. We have a description, and then we have a URL. And then it has its own primary key here as a resource ID. So this information is what makes it into the training portal. And the way that it does so is actually kind of interesting. I'm going to go and I'm going to show a tool that is not actually linked over here on the left. And the reason is you don't often have a use to go here. But if I go to the ontology tool, what this shows me are all the different quote unquote objects that are defined in the platform for N3C. And when I say objects, what I mean is what's possible to do if you have the right permissions to do so, and this isn't sort of widely available to do this because you can break lots of things if you don't do it just right, is you can take a table like this and you can lift the rows in that table to these quote unquote objects. And so here I'm seeing the training module object, and I can see various properties of them. And probably the most useful thing to show is actually how you would edit that property mapping. And so here I'm basically saying, listen, a training module object is defined from a row in this training modules data set, where the title, for example, maps to the title property of the object. And the description maps to the description property of the object. And you don't have to have everything mapped to a property of the object. Most things do, but not everything does. And these various properties of the objects, you can then go in and sort of give hints, right? And so I can say, all right, this particular description field is long text. That makes it sort of like searchable by other tools. It's a searchable and various things like that. Some properties you can say this is like the primary key for that property. So this is like the unique thing that says what that object is. The title field, you can say this is a title key. So by doing so, what we're doing is we're elevating the rows of this table into this sort of like generic object thing, and then annotating properties of that object with certain features of them. The other thing that you can do is when you have multiples of these created, so for example, I also have a training module resource object, which has a similar set of properties that we map to properties of the objects coming from our training module resource list. And then you can have links between these things. So you sort of define the fact that a training module and a training module resource are linked via a one-to-many relationship where the key is the module ID in both of them, and you define how things should be displayed when you're looking at the link itself. And once you've done this, once you've defined an object from a table like this, you can also do some really cool things. Let me go back to my training module. And we can sort of give a view of the object. So once you've defined your properties and your things about the properties that you're interested in, let me just pick one of these. I'll pick the knowledge store guide. You have this little editor where you can set up tabs in the view, and you can add widgets to the editor. And this actually is a fairly comprehensive way of putting user interface on top of object types. And you can even put like forms so that you can edit object types and permissions around who can edit the object types and what they can do with them and all that kind of stuff, which is kind of cool. And so you sort of set up the view for an object. And once you've done that, you can go to the Object Explorer. And this is something that everyone does have access to, which is the Object Explorer. And you can see all of the different kinds of data that have been lifted up from boring, data tables into these objects. And here's our training modules, and here's our training module resources. So if I were to select in the default sort of view of a bunch of objects, like a training module is just like some histograms, and I can see a list of them all. But if I click on a particular one of them, then I open up that object's view and I can see the description of it, and I can see the various linked information and so on and so forth. What's really cool about this, I think, is that by sort of taking a data set and then lifting it into this generic object framework that's defined by the Enclave platform, Palantir's Foundry, you can then inject it into lots of other tools in the Enclave. So code workbook templates, for example, operate on these objects, and you can use them in your code workbooks. Contours, you can use objects in different ways in contours, and it all comes along with the fact that you've lifted it up into this object definition. And so that's the overview of the backend object system. Now, what I think is especially cool about this from an N3C sort of community perspective is that if you're able to log into the Enclave, we already know some things about you, right? Like we know which institution you're from because that's part of the registration process. Once you've signed a data use request, that data use request form is actually interacting with this object system. And so if I were to go to the object browser here, I can find under the N3C admin section, and I can go and look at, for example, the research projects. And these are the objects that are defined in the research project. So it means that there's a table back there somewhere, right, that someone has set up the UI of what we would see when we look at a research object. I'm going to open one here, immunosuppressed. This one. And so this is the research project view. And as the Enclave has sort of evolved and other people have defined more objects, we've gotten more links between objects as well. So now we can see down below, for example, that this particular research project has 3 collaborators and has 1 OMOP concept set linked to it. And so that means that that particular project did that. And in fact, that wasn't the one that I wanted. This is the one that has many more things like knowledge objects that have been used by that particular project, knowledge objects that have been produced out of that particular research projects, all the researchers involved in that particular project, the various concept sets that people have linked and sort of said, yes, we're using this concept set in our project, and of course the data use request project object itself. And you can see in the various, title, description, various other things like that. One of the widgets that can be put into the Object Explorer is what's known as the knowledge graph. And in the knowledge graph, you sort of start with the object itself. And over here on the right panel, I can see underneath the object some sort of information about it, links to other resources. I could go to the overview to get a sense of what's going on with this object. But what's especially neat about it is I can go to the search. When I have this particular object selected, I can search for all of the things that have been linked by that linking criteria in that sort of ontology editor bit. And so I could say, you know what, I'd like to see all of the researchers that are affiliated and linked to this particular object, and I can search for it. There are 40 of them. And if I wanted, before I sort of add them to this graph, and there's only a certain number of nodes this graph can display. And so if you're trying to do like 200 of them, you might want to filter it down by adding a filter to say, you know what, I only want to see the citizen scientists, or I only want to see people who have signed a DUA, or so on and so forth. In this case, I'm going to go ahead and add the nodes to the graph. And that brings in all the researchers that have signed the DUA for that particular project. And then I can deselect and I can sort of browse these things around. What's kind of neat about this is you get a little bit of, given sort of nothing selected, you can see some summaries of how many object things you've got in your little graph, and I can change the color. So if I wanted researchers to instead be colored kind of a lightish greenish, I can do that. I can add badges. You can do a little bit of visualization stuff. But what's especially cool about this is I can sort of expand this search. And so I could say, let's actually select all of the researchers, all 40 of them that are in my graph right now. And I'll say select. Am I sharing my screen with you guys? Yeah, okay, so and this sort of selects them all, and the overview gives me some info about them. So it tells me that 2% of the people that I have selected have a name David Patch. That's always useful to know. Nobody's a citizen scientist. 20% are from Johns Hopkins University. And so it sort of knows this information. And with those selected, I can go back to my search and I can say, okay, I've got a bunch of N3C researchers selected. Let's actually click on N3C institution to see which institutions those particular researchers come from. I can do my search, and it's going to find 18 different institutions. I could filter them before I add them in if I really want to, but I'm not going to add them in. Now I get a view that looks kind of like this. It's a little bit messy, so I'm going to change the layout here to be layout all. Sometimes it helps, I find, if you do like a force directed and then you do a layout all, or vice versa. But now we can see that we have our research project. Oops. And there's a cluster of folks here who are involved in that research project from a particular institution, and it happens to be Johns Hopkins. Similarly, if I browse around, I can see the other institutions. Somewhere in here will be UC Denver Anschutz Medical Campus. That's where I'm from. And so what I'll do is I'll actually say, you know what, let's find out who else is involved with N3C from UC Denver, Anschutz Medical. So selecting that particular object, I will then search for N3C researcher, perform my search. There are 32, so I'm going to add them. And we've added a bunch of researchers to our graph who are involved in other projects potentially. Before I deselect these guys, because I kind of want to find out who is involved with other projects, I'm going to sort of drag this to the side and just leave them selected. I've got this set of researchers selected. From there, given that they're selected, I can say, you know what, show me all the research projects that they're involved in. $16 of them. I can add those nodes to the graph. Now, of course, it's getting quite messy. So what I'll do is I will redo my layout here. And we can see, for example, that from UC Denver Anschutz Medical Campus, there are a number of folks who aren't involved in any research projects. So these are folks who I might want to reach out to and say, hey, have you been having fun in N3C? You should come hang about, have fun in N3C if you haven't already. Tiffany is involved in a number of projects before. And then there are some folks sort of in the middle of these two projects who are involved in a number of different projects and a number of sort of key players in N3C. Peter DeWitt, Melissa Hendel, so on and so forth. So I just think that this is a really neat thing that we can do. And much of this is enabled by the operational data tracking that happens and the tooling that sort of comes along with it. And I just wanted to show you this one because I thought it was super fun. You can go in, you can do this. Again, it's the Object Explorer, and you can go find information about it by finding under Object Explorer, the N3C admin section, and you can check out the researchers, research projects, and somewhere in here is the institutions. But you're not limited to that either. I could go back to my graph. Uh-oh. This one. And I could say, given this particular research project that I knew I was interested in, this immunosuppressed one, what if I also wanted to see what concept set they're using? Or I wanted to see which knowledge objects they're using. Those are easy enough to add as well. And much of this does depend, by the way, on people going in and noting in the relevant tools, whether it be the concept set browser or the knowledge store, yes, our research project is using this particular concept set or is using this particular knowledge object, because some of that linkage information is not automatic. So I'll add 11 knowledge objects and 73 concept sets. Oh. There we are. That's all I wanted to show, just a little bit of a tour of some of the tooling that happens inside and something cool you can do with it.
Cool. Well, thanks, Shawn. Questions?
Hey, Shawn, this is Warren. That's really great. Do we all have access to being able to explore institutions and who's at institutions and parts of projects?
As far as I know, if you have access to the enclave.
Great, thank you.
That's nice. What do you see as the next thing on your roadmap?
Oh, I didn't build any of this. This is all the platform people. I think that there's a lot of use in this. I'm looking with one of our interns at concept sets. For one thing, looking at relationships between concept sets. The concept set editor allows you to find a concept set that's similar to the one you're currently editing. So you can look for like, oh, this concept set already exists. I'd like to see a little bit better tooling around finding connections between concept sets. I really think that if people go in and make links between their projects and the resources, whether they be the concept sets or the knowledge store objects or the community notes that was presented by Tim Bergquist is based on all this technology as well. The more connections that we make between all these bits of data, the more that we can do with it. And I think the more useful it will be for discoverability as well. Because I can easily, for example, take a concept set that has a number of projects, figure out what projects that concept set is used for, and then from that set of projects, find all the other concept sets that might be of interest to my research project, just based on thematic connection.
Yeah, that'd be really useful. That's very interesting. Question from Armin McKelly: who's the point of contact to request a new concept, e.g., new drug added to the concept set members table?
That particular one is user managed. You can create your own concept set, and we have information on how to do that in both the training portal. In this case, the Intro to OMOP and Contour one covers concept set creation, and we also have a community note, I believe, about it.
Cool.
The community notes application.
Okay, this is fantastic. Any other questions? Okay, well, this is great. Both presentations were really fantastic, and I thank both of you, and we'll give you guys 6 minutes back. So have a great week. Thanks.